Okay, today again I'll be continuing my last lecture, Industrial Applications, Basic Principles in Bioprocessing Technology, where we have already discussed about some of the metabolites like antibiotics. We discussed about the discovery of antibiotics, and then we also talked about the microbial reservoirs and industrial microbial product overview and principle of antibiotics, amino acid production, organic acid in food. Well, so we'll continue with the organic acid and food. What is the role of organic acid in food processing? Uh, Organic acids such as citric acid, lactic acid, and principally they are preservative for the food. So, which we already discussed in the earlier lectures. So, we'll proceed further after the citric acid, lactic acid, and the acetic acid use. What are the biopolymers? So, microbial plastics are basically one of the area which has been already talked about in my last lectures. Okay, so we'll start from here. The biosurfactant. Now, biosurfactants are the nature's detergents. Biosurfactants are basically the amphiphilic molecule with both hydrophilic and hydrophobic regions, and they basically participate in interference between the fluid different polarities, both the hydrophobic and hydrophilic polarity, and they make them available for emulsifications and detergency of the bioremediation. So, these detergents are basically used for bioremediations of various oils, various organic materials, and various fatty acids also will get dissolved through these detergents. Suppose to clean up the oil spill, essential for the dispersing oil spills and environmental remediation. So, whatever oil spills are being made can be dispersed out from through these detergents and bioremediation process, basically in the ocean and other places. The enhanced detergency is also attained through improved cleaning effectiveness and in various applications using these detergents, natural detergents. Recovery of the oil is also attained using the enhanced oil recovery from the underground reservoirs. So, all these processes is basically used through biosurfactant. Biocatalyst is another product like precision chemical modifications, where we use bio transformations to modify the steroids. So, as shown this example here. So, this is how conversion of this through Rhizopus nigricans. So, Rhizopus nigricans is basically utilize the compound steroids, and this is called hydrolysis of the progesterones to 111 alpha positions at the Rhizopus nigricans, and this Rhizopus nigricans is basically responsible as the microorganisms to convert them into. So, this is how the in the fungal culture we can convert these steroids. So, this is called kind of a chemical precision chemical modifications, how the chemicals are modified through these microorganisms. Basically, these molecules acts as a substrate. These molecules acts as a substrate for these fungus molecule, so that they can easily transfer them into other chemical modifications, right. So, typical bioconversion is the hydroxylation, where the hydroxyl group is being added like this, right, hydroxylation. And this because of this hydroxylation, that is addition of OH group, this is progesterone by Rhizopus nigricans. The steroid is dissolved in acetone before addition to the pre-grown fungus or fungal culture, and this is demonstrating precise molecular modification under the mild condition. Now, having these modifications basically make the chemicals are more useful for the microorganisms to be utilized for various metabolic processes. So, this is one of the advantage of this modification. As advantages of the biocatalysis is basically biocatalysis, again I repeat definition, that the catalysis of these chemical moieties taking place in the presence of microorganisms, or the microorganisms are responsible to catalyze these reactions, right. So, stereospecificity, then mild condition produce the single biological active isomer rather than environmental safety, reduces harsh conditions, need for harsh chemicals and extreme reaction conditions, then operate at normal temperature, pH, reduce energy costs. All these conditions are required for basically conversion of these microbial flora or microorganisms to convert these molecule different product through the catalysis, and that what we call the biocatalysis. Reverse vaccinology is another area where the mining genomes for vaccine. So here the whole genome of microorganisms are basically, it's a new concept you can say, where this whole genome is being used for designing a vaccine. Or basically vaccines are, we call the reverse vaccinology, where we can mine the pathogen whole genome sequences to identify the antigen that may be good vaccines, right? Or it could have a specifically targeting to the site. And this approach is very important and essential when the pathogen cannot be grown in a vitro or produce the molecule to similar to the host molecule. So the genome analysis is examine pathogen genome for surface proteins and secreted antigen. It can be antigen selection process where they identify the protein expressed during the infection and found in all strains, and then mouse testing where they test hundreds of candidates for immune responses generation, then clinical trials where the advanced promising candidate to human testing. So this is how the advantages of this technology for the reverse vaccinology. Now MAN-B, this is one of the vaccine development success story where the MAN-B vaccine is basically exemplifies reverse vaccinology success. How reverse vaccinology process can be used to develop a vaccine. So this is one of the case study. Because the capsular polysaccharides of the group B, Neisseria meningitidis, which cause the meningitis, is identical to human polysaccharides, and traditional vaccine approaches failed to nullify the effect of this bacteria. So using the complete genome sequence of 600 surface protein were identified and 300 tested in mice, leading to about 25 worthy candidate vaccines. So this is called epitope vaccines. About 600 surface proteins, then they are tested in the mice at 300 different proteins, and for getting the promising effect of these epitope these candidate vaccines to show the immune response is basically 25, which are important, worthy for the development of vaccine molecule. Now, the biofuel production, where the meeting energy demands, because biofuel is very important in order to get the energy and keep the reservoirs of the fossil fuels. So what we do, basically the new concept is global energy consumption. So we can conserve the whole global energy. So it is said that 71% energy growth is by predicted increase in the global energy consumption by 2023 through this method, and 10% ethanol content, like current ethanol content in gasoline is in the United States, and about 3 times energy potential by hydrogen gas, or hydrogen has 3 times more potential energy per unit weight than gasoline. So this criteria is basically this background. Based on this background, the renewable energy source is based on the microbial transformation, where the microorganisms involved in transforming either the natural microorganisms or the microorganisms which are designed or engineered through recombinant DNA technology can be used for viable approach to solve the global energy climate crisis. Now let's talk about the ethanol. Ethanol, the leading biofuels, because this is one of the most important biofuel used today, which are being used for many purposes. Ethanol is currently used as an additive to gasoline because it is easy to store and can be burned in car, designing to use the pure gasoline, right? And corn is currently the substrate for most of the commonly used for generating the ethanol. Even molasses can also be used for generating ethanol. So these are all waste product can also be used. So cheaper cheaper raw material can be used to produce these ethanol, and in many countries like in the United States, they've been using through fermentation process. And Saccharomyces cerevisiae is a fungus fungus type here, which are using to ferment the molasses or these ethanol production process. corn starch is degraded using amylase and amyloglucosidases, and these enzymes are utilized to convert these corn starch into the glucose, and the glucose is utilized by the Saccharomyces cerevisiae to production of ethanol. And that later on it can be distilled and purified in several steps. So what are the challenges with the corn-based ethanol? Because agriculture fuel is important today because they can have low cost and better fuel energy consumption or better fuel energy reservoirs. The use of corn for ethanol production has been unintended consequences, chiefly in the form of like higher worldwide food prices, and most researchers is devoted to finding other substrates like for ethanol, like molasses and some other waste materials, including crop residues and algae also. The food price impact. What is the food price impact? So the corn diversion to ethanol increases the global food cost. Energy balances, like some analysts suggest entire ethanol manufacturing process may consume more energy than product yield. Alternative substrates, like research focuses on crop residues, cellulose and algae as sustainable alternatives. So keeping that in view, we can plan to improve the economy of through ethanol production, and we are trying to use the best possible substrate, the best possible raw material for production of ethanol through microbial world or through microorganisms. Moreover, we can also improve the strains quality, or the high producing strains of these ethanol, like Saccharomyces cerevisiae strains, which can be used during fermentation process, and they can produce better quality of ethanol. Hydrogen. The ultimate clean fuel is hydrogen as a biofuel compared to very favorable to ethanol, so, and other fuels also. It has about 3 times more potential energy producer per unit of weight than gasoline. So ultimate hydrogen is the best fuel to be considered, then better than gasoline, which make the highest energy content and which is available in the fuel. However, the shortage and distribution remain insignificant, but the challenges are still there. Clean combustion. So you can also talk about how it clean the environment, right? So hydrogen burnt cleanly, producing only water as a byproduct. So there's no harm in burning the hydrogen in order to produce some kind of a pollutant in the atmosphere. So this is one advantage of this hydrogen. Then storage challenges is there. So it requires the specialized storage and distribution system, which is one of the major challenge for us. Microbial production, like diverse microbes produces the hydrogen through fermentation and photosynthesis. So we have to identify several microorganisms, rather strains of microorganisms which can produce better quality of hydrogen, or high quality of hydrogen, producing less contaminant or less pollutant in the after the biocatalysis. Microbial hydrogen production method, it's a diverse group of microbes producing hydrogen, making its manufacturing quite flexible. It can be produced as a product of fermentation by anaerobic microorganisms and by oxygenic photosynthetic microbes using 2 different enzymes, hydrogenase and nitrogenase. Fermentative production, photosynthetic production, enzymatic pathways. In fermentative production, anaerobic bacteria produces hydrogen during the fermentation process. Photosynthetic production, like cyanobacteria and algae, generate hydrogen using light energy. Enzymatic pathway like hydrogenase and nitrogenase enzyme catalyze the hydrogen production. So I think these 3 facets of the microbial hydrogen production is important. How we can produce the hydrogen and what kind of a microorganism are being selected for production of hydrogen. So ultimately we can identify certain microorganisms and the condition where the microbes can better ferment and better grow in the fermentation conditions, and acquire or adopt the pathways that can produce the better quality of hydrogen through their catalysis. either through oxygen, through photosynthesis, or through fermentation process. In both the cases, it depends on what kind of microorganisms you are taking, so they can consume accordingly. They can go through the pathway of photosynthesis like a cyanobacteria, or they through the fermentation like through other bacteria, bacterial process like anaerobic bacteria. Now, industrial fermentation system, whatever experiments are being done and confirmed through laboratory or even a pilot plant experiment, which need to be translated into the industrial production, because for industry production we are not talking about in terms of like a milligram or microgram, but we always talk about in like grams and kilograms of and liters and gallons of a product. Now we have hundreds of thousands of liters. Large products are being produced through the fermentation process, and for that we need to design the industrial fermenter. And designing industrial fermenter is again a big task, where you need to have the aseptic conditions, best quality fermentation conditions, where the samples can be given to the fermenters, and it can run oxic and anoxic, both the conditions, where it depends on whether you need kind of aerobic microorganisms or anaerobic microorganisms. So if it is aerobic microorganisms, the oxic conditions has to be maintained in the fermenter. If it is anaerobic, then of course anoxic conditions need to be made on the microorganism during the sampling processing. Certain biosensors provide the real-time information about the fermentation process, so you can use this biosensor to get the information, and these information are to be carried out through this. Then scale up from flask to factory, again from lab to industry. This is how the main task of the fermentation process or the industrial process, where the success of an industrially important microbial enzymes or biocatalyst product, where we need to scale up the fermentation process and we need to scale up the production. So at the laboratory scale, the initial development of like 202 up to a liter of fermentation, 2 liter of the product. So we can use like a simple flask, like we call Erlenmeyer's flask, or we can have like normal flask we are used for that, just to use the incubators, and even in the fermenters maybe 1 liter of flask can be used by small fermenters. But for the pilot scale, we need to increase and optimize the conditions, because when you increase the scale up the conditions, then scale up the product, then the conditions need to be optimized, because the same product which you are producing at a low quantity, like in liters or in milliliters, then if the scale up taking place, then you need to have change the conditions, or not change but to optimize the conditions. The industrial scale up conditions, like full production, is hundreds of thousands of liters of reactors are used with the maintained microenvironment. So the microenvironment is something which is basically the environment which provide all conducive environment or conditions, experimental conditions, better experimental conditions for the high quality and more quantity of the product during the fermentation process. So this is called scale up process from flask to factory, because in factory you have big fermenters and big flasks, gallons of vessel, where the substrates are used for productions of these product. The primary versus secondary metabolites. As we all know that the metabolites means the outcome or product produced by the microbial world or microorganisms. So the microbial product are classified into primary and secondary metabolites. Primary metabolites includes the compounds related to cell synthesis during the exponential growth, like in log phase, while the secondary metabolites accumulates when the nutrients become limited following the growth of the bacteria, and they check the growth of the bacteria, and then they can start producing from the stationary phase to onward. Now, for example, primary metabolites like amino acid, nucleotides, which are essential requirement for the growth of the microorganisms. Fermentation end product like ethanol, again this is also the result of the primary metabolites, and they can be part of the growth of the cells to get the energy. Industrial enzymes, several enzymes are produced through microorganisms, which can be utilized later on either through purification system or the whole microbial extract as it is. then produce during the log phase growth, right? These are all like primary metabolites that produce on the, so if you take a growth curve, it is all like in, so this phase is basically the primary metabolites. Now the secondary metabolites start from, as a result, from here to here, we call secondary metabolites. Most of the antibiotics and mycotoxins, they are produced as secondary metabolites. It limited the relationship to cell synthesis, because sometimes the cell synthesis is also stopped by producing the metabolites. When the accumulation of these metabolites takes place in the stationary phase, cell may not further grow, right? Accumulate during the nutrient limitation. When the nutrients are stopped, then they start producing the secondary metabolites, maybe for sometimes for energy productions, for sometimes some other reasons. And they produce after the active growth phase, that is in the stationary phase. The genetic manipulation for industrial optimization. What does it mean? Genetic manipulation for industrial optimization means how do you genetically modify, right, for a product produced. So suppose if you have a microorganisms, right, and this has some genome, and this genome is genes which are present in the microorganisms, these genes are responsible for the production of, for example, ethanol. Ethanol has certain quantity or quality. Now to improve the quantity or to enhance the quantity and to improve the quality, you need to manipulate at the gene level, right? And this manipulation of the gene level is called the genetic manipulation, where the rDNA, right, or we call the rDNA technology. We will talk somewhere in maybe some next lectures or some next slides, some few more slides, we have some rDNA technology. How this rDNA technology used to manipulate this bacteria to produce the better quality ethanol and better quantity of ethanol. So this is the main task. So industrial manipulation is basically involved in the production of the best quality product. Now, so one thing is that you have a natural microorganisms which are present like in environment. You are using, for example, Saccharomyces cerevisiae. Now this Saccharomyces cerevisiae producing the ethanol. So we have a one quality of ethanol, it could have some quantity of ethanol, but the same bacteria or same fungus, when they have got to certain mutations. So for example, they are talking about the mutation markers is producing. So this is all the graph showing the how the mutation makes it possible to increase the fermentation process. So we have to identify the point mutations, we have to identify certain mutations, and this manipulation are either through site-directed mutagenesis process, or through random mutations, or through specific mutations, or through spontaneous random mutations, and screen the best quality product, best quality microorganisms which can produce the high quality fermentation product or fermented product. So this is called the, so that increase basically the several tens to 100-fold of the product quantity, quality can be improved through genetic manipulation. Now this is what the site-directed mutagenesis is. So how you can modify it, for example, if you have a gene here and this gene is having A, T, G, C, T, T, for example, this area of the gene, if we modify this G to C and we call it as A, T, C, C, T, A. So by changing this G to C, the codon TGC is changed to TCC, and this change in codon from TGC to TCC make a new amino acid. So if in the protein you can change the new amino acid, that could have some new impact. So this is called the site-directed mutagenesis. Now the question is that you can ask a question like which amino acid to be changed. For that you need to identify the base codon, in which codon you need to have. So you have to go back from the required amino acid to be changed, go back to their respective codon, base to be changed, so the codon can be replaced. So all this manipulation is being carried out through site-directed mutagenesis process. Now in a site-directed mutagenesis, what is being done, a primer is designed. For example, if you look at this, this figure, So this is the plasmid where you have a target gene, and this target gene have a mutation somewhere. So what you can do, you can have a primer. Primer is some oligonucleotide sequences, right, 5 prime to 3 prime, 5 prime to 3 prime. Now this primer may have replaced one of the base T, G, C or whatever, and by changing this primer is being cloned after the amplification through PCR, and the product which is formed we call amplicon, having the mutation at that particular site, and that to be cloned, then that amplicon is cloned in the vector, and the vector which is produced finally is a mutated vector. So this is the method of site-directed mutagenesis, where you can easily modify a single mutation. Now the beauty of the site-directed mutagenesis is that through this site-directed mutagenesis you can specifically modify a specific region of the gene, and that could have an impact on the product. So accordingly you have to play around with the all permutation combinations that you want to change in order to produce the best product. And so you also have to learn which amino acid, which changes need to be made so that the product can have better quality. So all these study to be done through genomics and through some other background studies. Now directed evolution technology. In directed evolution technologies, what we call the error-prone PCR, where the directed evolution change is taking place. Basically all these technologies or all these techniques are being used to improve the microorganisms, to improve the microbial or microbes or microbial world in terms of like bacteria, fungus, so that they can involve in the fermentation process and produce the best quality fermented product or fermentation product or good quality fermented products to be produced. So besides site-directed mutagenesis, you can have a directed evolution technology, where the directed evolution technology means rational approach to for the production strain. You can have a construction by targeting only gene of interest, like specific gene if already known for mutagenesis, and several techniques like enable large scale changes in the gene beyond the simple directed mutagenesis. So you can have a lot of small changes, deletion, insertions can be made. So accordingly you can customize the gene product. So error-prone PCR, we can use error-prone DNA polymerase to create a gene, gene variants using amplification, DNA shuffling, randomly fragment similar gene from the different species for recombination process, whole genome shuffling, recombine entire genome using the protoplast fusion techniques. So this is all being done through the new technologies. CellX is a new technique we call the engineering RNA therapeutics. So what does it mean? It means this is the systematic evolution of ligand by exponential enrichment. So keep including the the product quantity, product quality keep maintaining, and the quantity keep increasing. So we call the systemic evolution, evolving in the best way, the best possible product of ligand by exponential enrichment generates libraries of up to 10 to the power 15 different RNA molecules. So it's like trillions and trillions of different, even hundreds of trillions of different molecules are being produced. And this process creates aptamers, small aptamers like RNA ligands, and that bind tightly to the specific targets molecules for therapeutic applications. So these are design molecules, and these designs molecules are basically for specific genes which need to be targeted. So this has been used for various protein which are like a disease forming proteins, if you want to block these proteins. Sometimes you can also use this process for expression of certain protein through promoter regions to enhance the process. So lot of manipulations at the genome level can be done through these aptamers, and these are in huge numbers. You can produce these RNA ligands, you can bind to a specific RNA molecules. So the sequence is already known to you before you design these aptamers in order to form them to a specific site. Now the metabolic engineering modular biosynthesis, this is another area where you can have modulate some molecules, some chemical molecules, and these modular assemblies of the different chemicals produced by the bacteria or fungus can be utilized by several microorganisms. For example, anti-cancer agents like opothiolone C and D synthesized by the modular enzymes complexes. So these modular enzyme complexes can be used for the assembly process, and they can produce the each modular catalyze specific reactions. It mixing the domain from different complexes can generate new products, and these products are basically utilized by the certain other for other uses. In agriculture, biotechnology, the tie plasmid engineering. So this is a very interesting area where you can talk about the transgenic plant, and these transgenic plants are basically nothing but you modify the plant. For example, if you want to make a virus resistant plant, if you want to make a the herbicide resistant plant, if you want to make like a disease resistant plant, so the gene to be identified and put in the tie plasmid. Now having the tie plasmids, if you look the figure on my right side, the tie plasmids which is quite big in number, like 150 to 200 and kilo base pair size of plasmid. It's a very huge plasmid. So one of the major challenge handling the tie plasmid is the transformation. Transformation is not possible. So having known that how it being transformed is basically what you have to do is basically to cut this in by we call this disarming. So you can engineer this tie plasmid by disarming, then adding certain gene and make the co-integration or binary system to be used in order to transform it. So all this processing, that is co-integration method, binary system to be built in, disarming, all together they make this tie plasmid better vector to carry the gene of interest to transfer through tDNA and virregulon, which are part of the tie plasmids, into the plant system through so this process we call Agrobacterium tumefaciens or Agrobacterium rhizogene mediated gene transfer. Now this gene transfer is basically more efficient and safe, and generally all the transgenic plants are made through this method. So tie plasmid engineering is another area where you can engineer the tie plasmid tDNA for transferring the plant through tumor inductions or through root inductions, as the case may be. If it is tie, it may be a Rhi plasmid. There is one Rhi. Rhi is basically root inducing plasmid. Tie is a tumor induced plasmid. The function is same, but the mode is different. Now this is how the Bacillus thuringiensis is natural pesticides. Now Bacillus thuringiensis is basically Bt toxins, and these Bt toxins are produced from one of the bacteria, and these Bt toxins are produced from one of the bacteria where it can be used as a insect resistant plants, and insect consume these Bt crystal, we call Bt crystalline toxin protein with plant materials, and they get lysed or lost. So insects can be killed, cannot be attracted by the plant, or if it is attracted, then it can be killed through making the transgenic plant by Bt gene or Bt toxin like a cryogene. So Bt gene is called crystal C cryogenes, cryoproteins. So these cryoproteins have a different kind. There's a two different categories of cryoproteins: low molecular weight cryoproteins and high molecular weight cryoproteins. And these cryoproteins are being utilized by making these Bt gene clone into the vectors into the plant systems, and the plant, when the insect ingest. So the beauty here is that these they have activation of this toxin in the alkaline conditions. So alkaline gut conditions of the insect hydrolyze these cryoproteins, and they cause the lysis of the insects, right. So this is one of the mechanism. They form pore formations takes place, and these pores basically integrate to the membrane, forming hydrogels and hydroxo hexagonal things, and then this make the like a lysis of the cells, and the water is ingested inside. Uptake of water takes place, and the more and more water is uptaken, then the cell get lysed and the insects get lysed out, and cell death takes place. So this is the mechanism of the death of insects through Bacillus thuringiensis natural pesticides. Now the microbial microbes as a product. So nanotechnology application is another area where we used to use nanotechnology like diatoms nanotechnology. Nanotechnology products are the natural, it's not a natural product, but we can use several plant products to reduce it into the nanoparticles, and the concept here is that these nanoparticles are more prone and more easily penetrated into the cells, and they can kill various microorganisms, or they can have more surface area, surface oxidation area. So it has also been explored in order to make different bioproducts. Bioproducts which are produced for certain use can also be used as a kind of a diatoms in nanotechnology. They can mix with the nanotechnology also. Okay, thanks a lot for this, and now this lecture is over. So we'll start another lecture soon